broadcast in a row now, part of the 7-part epic March Mammal Madness Festival. And so I'm going to change up the intro a bit by saying that with that countdown music at home, you are encouraged to dance along. Now, Jessica danced along with me at the beginning of this, and so she gets first dibs on questions because of her enthusiasm with our opening. Now, welcome in, everybody. This is our 7-part epic festival with March Mammal Madness. It is the 10th anniversary of this epic contest, breaking down epic battles between all sorts of incredible creatures, the occasional lichen, and all sorts of amazing things around this planet. If you guys are joining us for the first time learning about this, I really encourage you all to head to the LibGuide. It is your one-stop shop for all the information about the program, how you can get involved, all the cool educator materials, no matter what grade you're teaching, lots of great stuff there. Now, I am with Exploring by the Seat of Your Pants, and we are all about bringing conservation, adventure, and science into classrooms around the world through free virtual broadcasts like this. And frankly, it never gets better than today. Today is one of our biggest festivals we've ever done. It's been so fun already to start off with a sort of retrospective of 10 years of March Mammal Madness. An hour ago, we did animal behavior, which was a lot of fun. We got to learn some amazing scientists going out to the field, talking about all their wonderful experiences picking ticks and leeches off themselves as they beat through the bush to get a fleeting picture of some bird or mammal. I mean, it sounds like fun, maybe, we're not sure. But we're going to dive in today in our third broadcast with some real science, okay? I mean, people just walk through the bush, that's fine. They set up a camera, they walk away, they call it science, and that's cool. We appreciate that very much. I'm an animal behavior person by background, so I really do appreciate it. But in our third program, to continue the fun, we're going to learn about genetics. We're going to learn from some amazing people whose science takes them on a deep dive into the inner world of some of the creatures that they study. So I'm so excited to dive in. I hope you are too. If you're joining us on YouTube, let us know where you're joining from. We'd love to hear from you. We'd love to hear your questions. We'd love to have you talk smack like people have been doing throughout the entire day's festival. Now, without further ado, over to Jessica Light, Dr. Jessica Light, joining us at Texas A&M. So, Jessica, thank you so much for joining us. Thank you for having a big Texas poster behind you to sort of be very stereotypical and to commemorate your state. And take us away. What do you get to do? I'm so curious.
Hi, everyone. I'm Jessica. I'm an associate professor at Texas A&M University, and I define my research as kind of centering in evolution and trying to use genetics as a tool to better understand how different populations and species are related to each other, of course, with a focus on mammals, but I also work with some invertebrates as well, especially parasites.
What kind of parasite? Okay, so we've already had ticks come in today. Is there like a specific thing that like gets you very excited? Tapeworms, flukes?
I got my career started with, well, leeches initially, and then from there I transitioned to lice, which solely parasitize mammals as well as birds. And I've worked with both chewing lice, which kind of chew on the skin and hair, at least of the animals that I work with. But I also have worked with sucking lice, including our head and body lice that many of our listeners may have personally experienced. I know I have, in terms of having head lice or a child with head lice in my own household. So I have studied them genetically to better understand their populations and relationships.
The sort of animal interaction that none of us really want to have, but it's very, very, very cool. By the way, on the leech note, because we've talked about this in 2 broadcasts now, we talked about genetics of leeches. Actually, I'll bring you up for this. One of the coolest pieces of science I've ever heard of in my life was done in Madagascar, and it was a scientist who went and studied the leeches because some of the lemurs, some of the mammals there are really hard to find. And so she'd harvest the leeches and get the DNA out of them to find out what lived in the forest, which is like mind-blowing and so cool. Like when you were doing leeches, was it for the genetics of the leeches themselves? Was it what they ate? I'm just curious.
It was for the genetics of the leeches themselves, but I think that whole idea of trying to assess the gut contents genetically of those leeches, because sometimes it's like an easier way to determine what other organisms are actually out there cohabitating with those leeches when they themselves may be really stealthy for those that are doing the behavior observations and not able to see them. But you can check out those gut contents of the leeches and get a good idea of what is out there in the vicinity that those leeches are feeding on. It's cool, it's really cool.
We started off with March Mammal Madness, and now we've gone down the leech train, which is very exciting. We're going to bring it back a little bit to mammals with our next speaker. So Dr. Eduardo Amorim, tell us a little bit about what you do. You get to do some really neat stuff. I'm very excited for our audience to hear about it.
Thank you so much. I am originally from Brazil, but I'm currently in Los Angeles, in California, and I am an assistant professor of genetics at Cal State Northridge. And my research is mostly focused on humans, but I also study how humans interact with other animals, specifically dogs. And I usually, in great part of my research, I get DNA from bones and teeth. We call it ancient DNA because we can actually recover DNA from humans that lived many, many thousand years ago. Currently, I'm studying the South American indigenous peoples that inhabited the high altitudes in the Andes, where oxygen is really low. So I'm trying to understand how adaptation to this environment that has really low levels of oxygen happened in humans.
So one of the themes that's sort of been pervasive today is that scientists get the chance to work with some of the coolest people around the planet, and they get to go to really cool places if you want. So kudos to you for coming up with a study topic that allows you to just go to Machu Picchu. Like, oh, I gotta go do some science. And where am I gonna go? One of the most iconic places on this planet. Very, very cool.
Cool, yeah.
Eduardo, so you talked about some ancient life. So DNA, okay. We've all watched Jurassic Park, hopefully. If you haven't watched Jurassic Park, leave this broadcast immediately and go watch Jurassic Park because it is a masterpiece. So in that, we have DNA that lasts 65 million years. How long does DNA actually last? Like, I think Jurassic Park might have milked it a little bit. How long is the DNA that you're able to work with?
I would have to Google, but as far as I know, I think Anne can confirm that, but it's, I think the oldest one we have is the mammoth. But I don't know exactly.
Yeah, and you were name dropped. You have to confirm. How old?
She's one of the
about a million years. About a million years, yeah. And yeah, horses and mammoths are the data that we have is older, the oldest.
Cool.
I work with a more recent time frame. My oldest, the individual that I studied, is the oldest is 8,000, 9,000 years old.
Yeah. Very cool though, and a beautiful segue into our next speaker. You're the first person to do that, so gold star to you. And so, like, why mammoths? Why horses? First of all, we'll dive into your topic specifically, but like, is it just because we have lots of samples of those creatures that we're able to extract that much DNA, or is it something unique about them? What's going on?
Well, you know, they lived in a really cold climate, and so often when they died, they got entombed in permafrost, and so that's like being in the freezer for, you know, a million years or 50,000 years or 500,000 years. And so it's kind of the perfect place to preserve DNA. So a lot.
I'm going to share a gross story before we continue on with learning about what we do. And that is, we have a speaker that comes on regularly on our broadcast. His name is George Brunis. He's one of the big iconic explorers in the world right now. And he tried mammoth. It was frozen, and he ate some. And he said it was absolutely beyond disgusting, and he got really, really ill. But he's probably one of the only people ever to eat a piece of mammoth, which is something that you can do in some places in the world. Eduardo, that's the appropriate reaction. We got some scary faces. YouTube, you guys want to chime in on that by all means. But Anne, tell us more about what you do. You were telling me before we got on the broadcast. It sounds very cool.
So I'm a professor at Arizona State University, and my research is sort of at the intersection between evolutionary biology and anthropology. And so I use ancient DNA and modern DNA to study genetics and genomics of humans, non-human primates. I'm also work quite a bit on ancient pathogens. So particularly tuberculosis, the pathogen that causes tuberculosis and the one that causes Hansen's disease or leprosy. So I'm interested in how people's movements have fostered the movement of those pathogens. And then occasionally I've worked on some fun projects involving like dire wolves, which is another Pleistocene species. So that was really cool. Yeah.
They didn't put dire wolves in this year's contest, but dire wolves in any bracket. There's nothing that beats a dire wolf. First of all, it's the greatest name. It sounds menacing. Like, it's like wolf's already like, okay, that means business. And then dire, I mean, your chances of surviving are dire. Now, Katie and I were talking in advance of you coming on today. We didn't get the chance to talk before this broadcast, but like, you talk about pathogens spreading around the world. Now, people do this. We understand this. We get on a plane, we go, we bring a pathogen somewhere. Do animals do this as well? And how do they go about it?
Yeah. Well, they do. And actually, animals were what brought tuberculosis to the Americas prior to contact. We think actually that it was South American seals who had interacted with seals around Antarctica, and tuberculosis probably transferred from Africa, from animals there to seals, and then the seals interacted, you can kind of picture this, all the seals hauling up on an island together and barking at each other. And it's a great way, of course, to pass a respiratory pathogen. And so then it jumped from seal species to seal species and to the South American seals. And then humans are hunting seals and interacting. And so probably got it through eating contaminated meat, seals that had tuberculosis. So that was a finding we totally didn't expect. But yeah.
In pathogen research, I must say, as an animal guy by background, bats always get that bad rap, rats get that bad rap, and it's been the seals this whole time. You go to the California coast, they're all just like chat with each other, and who knew? Okay.
Well, Northern Hemisphere seals don't have TB. It's only Southern Hemisphere seals. And and we also learned that squirrels in the United Kingdom carry leprosy.
I'm from Toronto originally, where there's like 8,000 squirrels for every person. That's terrifying to hear. And by the way,
watch out for the squirrels.
We're not going to let any of the southern seals play in any reindeer games.
That's right.
So, thank you for that, Anne. Our fourth scientist, Elinor Karlsson, come on in. Tell us a little bit more about what you do. And again, YouTube class, if you guys want to start sharing questions about genetics, anything you ever wanted to know, no matter how nerdy, no matter how strange, we'd love to hear from you. But Elinor, take us away.
Thank you. So I'm Elinor. I'm a professor at the UMass Chan Medical School and at a place called the Broad Institute. And I study something called comparative genomics, which basically means that we've gotten really, really good at looking at the DNA of lots of different species. But when you look at DNA, you get this long string of letters out of A, C's, T's, and G's, and we're not very good yet at knowing what any of it means. And so we compare different species to each other and try to figure out what the DNA means. And it turns out, like Eduardo was talking about the high altitude adaptations, it turns out it's not just humans that have adapted to high altitudes. If you look in different types of mammals, you actually find the same gene, and each of them has changes in it that allow species that live at high altitudes to live at high altitudes. And so it's actually a lot of fun. You get to kind of think about how humans are special in some ways, but other species do some pretty amazing things that humans can't do, like hibernate or things like that. And so, and I have to say that I do have a particular interest in a species that everybody knows really well, which are dogs. And so we do a lot of work studying dog behavior and trying to figure out what changes in that very long string of A, C's, T's, and G's means that a dog becomes obsessed over retrieving balls. I just kind of think that's kind of amazing that can happen.
So, okay, here's the question on behalf of everyone who's ever wondered about genetics, and you get all these like newspaper headlines that are like, a gene for this. So when you're talking about these adaptations for like, okay, so we have this gene now, and it allows us to breathe better at higher altitudes. How do you find that? Like, what are you even looking for? Since it is just it's not like there's a section of the genome that says like, this allows me to breathe better or bigger lung capacity. So like, Eduardo, we'll bring you back in for this too. Like, how do you know?
That would be super helpful if we could just label it, to be honest. It'd make our lives so much easier if it was just like, this is the gene that does this. But no, it's not that useful. Eduardo, sorry, I interrupted you.
No worries. Yeah, so it's when we are lucky, we find the gene that is responsible for that. But usually there are many genes that contribute a little bit for the trait. Sometimes it's difficult to go, sometimes it go either way, So, but when it's one specific gene that it's involved, sometimes it's easy to compare a population that it's under that condition versus a population that it's not living in that condition. And then you can see if the sequences of DNA change and how they change. A little bit more complex than that, but it could go that way. But you could also make like a cell culture, like human cell culture, and you could change the gene. You could knock out the gene and see how the cell behaves. With oxygen, it's easy to do. But with other phenotypes like behavior, I don't think it's that easy to do.
And I think it's actually...
Go ahead, Elinor. Sorry.
I was just going to say the other thing, Jesse, that sometimes it's really hard to talk about, to describe it properly, because people think you have a gene for living at high altitudes, and that's not what it is at all, right? Everybody has. So there's one gene that's super involved in whether you can live at high altitudes or not, called EPAS1. Everybody has a gene called EPAS1. Yeah. I think every mammal has a gene called EPAS1. It's not that some mammals have it and some mammals don't. What happens is that some individuals have a change in that gene that lets them, is it carry oxygen in their blood better? Is that what it's doing, Eduardo? I'm trying to remember.
Yeah.
Yeah. So it's like, and so they're just a little bit of a tweak. You don't want to change it too much, because if you change it too much, you might break the gene, and you definitely don't want to break the gene. Super important gene. You just want to make it work better.
Okay. Unless you want to do some experiment with the gene so you can break it and see how bad the cell survives the condition. Yeah.
You guys
cell
are like,
culture.
you are like gods with your cell culture. So you do anything you want. You have all this power. Eleanor's like, yes.
Yeah. Or the soup is cells usually win, to be honest. Scientists spend a lot of time like just trying to figure out how to get cells to do something they want. And mostly the cells just do what they want. And every once in a while they'll go along with it and do what we want.
So I'm gonna bring up Jessica in a second here. So I love that sentiment, like this idea that, you know, as scientists, we do have a lot of power, but like we are constantly fighting for this. One of my favorite things in bacteriology is that like most bacteria will not grow in a petri dish. Like 95% of them will like never, they refuse to go in a little cultured dish. And so we have this really, stilted. I don't even know the word. My English is failing me because I've been up since like 6:30 this morning for how that exactly works. So Jessica, I'm curious, we can, like, again, it's a little mind-blowing. It's hard to fathom. Again, unlike those animal behaviorists with their cameras, it can be tricky stuff. So how do we even know, so EPAS gene, so we have this gene, we have this specific thing that we know is related in some way to this trait. How do we know that? Like, how do you ever find that out to begin with? Is it that you see that it's different in creatures that live in high altitude? How are you looking at that? Because there's so much, the genome's so big. So
yeah, the genome is really big. And this is sort of beyond what I generally do in my research, but I think Eduardo hit it, you know, right on the nail, on the head of the nail, and saying it's comparative, right? And you're also looking for some signatures in maybe how that gene might be behaving. Maybe it's under selection, natural selection or something like that. And there's some statistical ways that we can see if that's happening. But if you have other populations and multiple individuals to look at, then over time you can start to see these patterns and these differences that start to point to, oh, something's going on here. And nowadays, genomics and genetics is becoming bigger and bigger and bigger, and there's a lot of comparative data out there. So depending on what you're working with, you may not be starting at scratch with nothing in terms of comparative stuff. But other times you might be starting kind of in a new population with something that no one's ever looked at before, and you just get as many individuals as you can, and you try to compare those to each other, and then try to expand out to things that you think or know are closely related, and do some more comparative analysis.
Very, very cool. Thank you, Jessica. All right, and so I'm curious now. So when I was a kid, I feel old now, geez. Talking about Human Genome Project, it was like $1 bazillion, and we could like sequence one genome. Craig Venter, it's very exciting. And so we do all this, it costs so much money, and now I'm getting the sense there's like more genetic data than we know what to do with. So how do you sift through that? Like, how do you find, like, I guess there's just such a multitude of questions to possibly answer. What jumps out? Like, what are you diving in on that's really exciting that this sort of new world of easy sequencing allows you to do?
Well, I mean, we used big computer clusters is what we end up using to analyze that many genomes. So for tuberculosis, you know, when I'm studying the ancient TB genomes that are several hundreds or thousands of years old, I need to compare them to modern sequences of different types. And now there's over 10,000 tuberculosis genomes in the database. So it's really amazing. And it helps us really understand the geographical distribution of these tuberculosis strains around the world, and also look at how the strains might act under different conditions, in essence, you know, different transmission possibilities. So it really lets us, have a huge data set and great statistical accuracy at asking some of our questions.
Fantastic. Yeah. All right, we've got our first YouTube question, which I'm going to send Eleanor's way, and then we're going to dive in on some March Mammal Madness specific genetic extravaganzas. So our first question, and Andrew, who's been joining us all day long, nothing better to do at all. How easy is it to find shared data on known genomes? Like, where do we look for this stuff? Now, you guys are scientists, so you have access to all sorts of fancy places that you can go look for things. Is it easy for you to find this data? Is it easy to collaborate? Is it a field that allows for that? Is it something that people on the street can go and like look up genomes and stuff and what's going on?
Yeah, so as long as you're not working on humans, it's actually usually super easy. If you're going to publish a paper these days in genomics, you usually have to put your data into a public repository where anybody can access it. And one of the biggest ones in the United States is run by the National Institutes of Health. It's called NCBI. And you can just Google that and it'll come up and there'll be a site that talks all about all of this genomic data and where to find it. The biggest problem is that, as you just mentioned, the files get really, really big really fast. And so it's still really hard to analyze that data. And I think that's probably the biggest challenge that we're dealing with right now, is that it's not enough just to let people get to the data. We also need to give them an opportunity to actually analyze the data. And so we're sort of working. So lots of people are working now on getting analysis in the Google Cloud and things like that running. But genomics is one of those fields where the technology is changing so fast that we're kind of like spend all of our time like building the car while we're driving it. And so it's a sort of mess right now. It's like, oh my God, we needed this yesterday. Let's make it. So
I like the honest assessment of the field. Thank you for that. I mean, fun today. Jessica, I want to bring up, you're bringing up something on screen share. Was it something related to what we were chatting about or just getting stuff ready?
I was looking for the website for NCBI so that you could have it. And then I figured, folks can Google it.
I'll
Google
get
NCBI
people in the chat, I promise. But that is very cool. It's nice that that's an option. And of course, the reason that we don't have people's data is for privacy's sake, correct?
yeah, you can get it. It's just that you usually have to be at an official research institution. You have to submit a proposal and tell people what you're doing with it, because it's obviously much more secure.
I think our audience will be happy to hear that amidst all the 23andMe things in the world. Okay, so Jessica, actually, I'll come to you for this next bit. March Mammal Madness is upon us. You were a viewer in some of our earlier broadcasts. So, like, how does genetics relate? We were talking about the ecology of various species impacting some of these battles that are going on from a genetics lens. In the past, maybe some of the things that we've had in March Mammal Madness number 9 or 8, or this year specifically, maybe you had a chance to check out the bracket. How does genetics play a role?
Well, this is a tough question because I think it gets at some of the things that we were talking about earlier. Sometimes some specific populations, which we are definitely looking at for March Mammal Madness this year, might have some specific adaptations that are under genetic not necessarily control, but genetics that at least starts the route for those adaptations for those populations. So it's possible that for a specific group of individuals or a population within a species, they might have something different going on in terms of their genetics compared to other populations that might put them at an advantage depending on their competitor.
Very, very cool. Thank you, Jessica. All right, Eduardo, we're going to take you this. You got to take a stand here, okay? We're at the halfway part of our broadcast. Our other scientists have been at varying degrees. I think in our last one, they all took a stand. In the first one, none of them wanted to. But here's our question from Ms. Juricic's class. Which mammal do you think is going to win? And it doesn't have to be a mammal. We have some non-mammals. So if you want to throw the March Mammal Madness thing out the window and pick like the Dobson fly, you can. But like, what's your pick?
Just to make it clear, I don't know who's going to win. We don't have this information yet, but I think it's going to be the Pride of Lionesses. Right. I think, I don't know, but I'm rooting for the gray wolf, which is my favorite species. So I'm between those two. I don't know.
Well, you definitely stuck with mammals. For what it's worth, Anne in our last broadcast wants the lions to lose immediately to the moles. And so, like, you and her can fight that out later, but we have, like, some real lion haters, and you're going for it. Number 1 seed, by the way. So it's not a big stretch. But I wish you the best with the gray wolves. Wolves are awesome. I mean, we talked about dire wolves a little earlier. Let's bring in Anne. What do you think? Okay, Eduardo's team lion/wolf, very predatory mammal. What do we think, Anne?
I don't know. My cats would be for the glaring of cats, right? But and I always love the primates, so I love the lemurs. But I don't know. Grizzly bear is pretty
metal. Grizzly bear is pretty up there. I remember I used to watch Colbert Report when I was younger, and like they'd have like the threat down of the biggest threats in the U.S. and bears were always number one because I think they called them godless killing machines. Not strictly true. Bears are wonderful and cuddly and eat a lot of bear meat. But okay, good to know.
Yeah, and hippo's not in it this year, so you know.
Hippo wins.
Danger.
Yeah, like we like to stress. Okay, everyone's like, oh, hippos, they're so nice. It's like, the Nile crocodile, and like the most
number one killer of people in Africa is hippos.
Mosquitoes win, and then hippos, and like Nile crocodiles run away when the apex reptilian predators are like, oh, I don't want to deal with that thing. Very, very scary. Okay, hippos, they had to remove it because it was too intense. It was too powerful, I think. Awesome. Thanks, Ken. All right, Elinor, do you have a pick? What do we think?
Well, the one I want to pick is, it's not very high seated, but I'm going to go for it anyway. That New Zealand short-tailed bat, because honestly, first of all, my mother's from New Zealand, so I'm biased. Okay. And second of all, until people showed up, the only native mammals on New Zealand were bats, and so those mammals survived in a world entirely filled by non-mammals. So I feel like they should have an up there. Maybe not in March Mammal Madness though.
I think you know what happened. So all the tuberculosis-filled seals crossed the Pacific, and they tried to get to New Zealand, and the bats just beat them off, and that was the
No, they got there.
No, the bats tried their best. Okay, fair enough. Okay, so if they lost to the tubercular seals, that's not good, but good to know.
I should have rephrased that. They're the only terrestrial mammals.
Okay. You are our only. So Allison wants to know bats. We've got a lot of team bats. We have a lot of bats this year in all the categories, which is very exciting. So if you guys.
A lot of mammals are bats, though. I can't remember exactly what percentage, but a huge percentage of mammals are bats.
Yes. So there's I want to stress, there's a lot more of other groups of animals in the world. There's 300,000 kinds of beetle, which is ridiculous. My favorite line in nature is that God had an inordinate fondness for beetles. But mammals, there's 5,500, but 1,200 of them are bats, and no one knows this, and it's crazy. And then mice. So we had Patrice earlier talking about mice. They're very cool. Jessica, what are you yelling about?
I'm yelling about the bats because they're the second most diverse group within mammals. And yes, Jess, you're right. It's rats and mice. That's the most diverse group of mammals out there. So if I got to pull my pick in, I would love to go with my heart rocket. I'm going to go with some of these rodents, man. And I'm so glad to see the bats as well. But I'm kind of torn between 13-line ground squirrel because they're really fun to watch. They're kind of goatees. They just kind of bop around and eat. And then I also really like the prairie dogs as well. And I've definitely picked and passed rodents like porcupine to go all the way. And I've been sadly disappointed by my love for rodents.
You geneticists are going for the lowest seated things of anybody. I don't know what it is about this field, but all the others are Eduardo is lions and wolves, and he's out. But the rest of you are, and really you put it on the line, which I appreciate. By the way, these are our the YouTube is amazing. So, okay, if we had to choose, since 20% are bats, Katie's talking about 20% are bats, which if it's 40-20, so of the 5 of us, and you can vote on this on YouTube, which one of us is most likely to be the bat in disguise? We're not sure. Mark Pistol's coming in with Team Leaf Slug. We've got a bunch of slugs. This is very, very exciting. Geez, Allison, 1,453 species of bats. You guys are such nerds. I like to, Eduardo, you mentioned Google. Google's great when we're coming about talking about numbers of species and what percentage of our DNA is related to things. That is your friend when we're doing these rapid fire broadcasts. So that is fantastic. Okay. We've gone down the rabbit hole. Bat squad protected from tubercular seals. Tubercular seals are getting a very bad rap in this genetics broadcast. Didn't see this coming. See, this is half the fun of this festival. Okay, so we've got students, a lot of students that, again, get inspired by you awesome, amazing scientists, and they want to go into these fields. You've got teachers that are really keen, they end up following you. So, for kids that are interested in genetics, what would you recommend? What are some resources they should check out? What are some things you guide them to? Elinor, I'll start with you. What jumps out that we can get people super excited about genetics? And Eduardo, I'm coming to you next.
I'm going to be super self-serving because I have a community science project and I actually want to know all about your dog. So, you can go do do dog genetics at our community science project, and we really want to know all about their behavior, because we want to figure out how. So basically, genomics is really, really, really complicated, and the data is really, really, really big. And so we need lots and lots and lots of dogs to participate in order to find things in their genome. And so we're trying to have resources on there where we talk about what we do with the genomic data and how we figure out what breeds are in dogs and things like that. So that would be one place to check out. But it's interesting because I think I'm not as up on all of the educational materials out there in the genetics world. So I want to hear what other people think.
Yeah. Eduardo, come on in. What do you think?
I'm not sure if I got the question correctly, but I think genetics, the good thing about genetics, in my opinion, I have currently my first students. I just started in August as a professor. So my students are now, and the nice thing about genetics is that you can either go through like a more laboratory route or a more computational route. So there is anything for everyone. You know, if you like more mathematics or more computation, if you want something more like bench work, working with like pipettes and doing some experiments, so on, you can do that as well. So I think genetics can go everywhere, and you can also use genetics to answer to ask questions about evolution, about diseases, about history, about anything. So yeah.
That is a very beautiful answer. I like that. Actually, since you've given diverse answers so far, I'm going to take the question from all of you. Jessica, anything that really jumps out to you?
What jumps out to me, instead of trying to answer your question, is to maybe let all the students out there know that genetics might sound kind of scary, right? Just the word that it is and maybe what's involved with doing it. But at the end of the day, it's a skill to help us answer questions that we're interested in. And just like Eduardo said, there's like multiple skills that you can use, whether it's at the bench, which is just like cooking, but just really small quantities most of the time, or it's at the computer. These are all skills that you could learn to help you ask or answer those questions that you might have about, you know, the evolution to adaptation at high elevations, or the evolution of particular coat color for fur or feather color for birds. And genetics is just a tool, and one of many tools that we can use to try to answer our questions.
That is a beautiful answer. And you're 3 great answers ahead of you, so no pressure at all. But like, wow us with your answer. I'm curious. Here, I'll unmute you.
There are a lot of online resources, right? So, for example, there's the Cold Spring Harbor DNA Learning Laboratory that has virtual exhibits online, and there's a terrific exhibit at the Smithsonian on outbreaks, and right, talking about the different ways that you can investigate an outbreak, and that includes genetics. So, really, genetics is a tool that can be used by kind of all corners of biology and anthropology to answer questions. So there are a lot of great resources out there.
Fantastic. We talked about online researchers. We talked about all sorts of great stuff. I must say my own interest in genetics was definitely sparked by books. I love books. Some of these books are literally behind me right now. 3 that jump out that I read when I was younger. The Gene, Siddhartha Mukherjee, Selfish Gene by Richard Dawkins, Genome by Matt Ridley. Fantastic reads, got me into the field. Again, there are more modern analogies of this for some of our younger students, certainly. One book I'd also really recommend, sort of on the really exciting world of CRISPR right now, which a lot of our students might know, The Codebreaker by Walter Isaacson, about Jennifer Doudna and Emmanuel Charpentier. Like, just an incredible story, something that like really is going to shape the world in the next 20 years in a lot of ways, which is one of the exciting things about having a bunch of geneticists on today for this broadcast. All right, okay. Anne, I'm actually going to take this right back to you, as we've talked about seals a lot. So, Anne, we'll bring you up, and then we'll bring up this question. Ms. Juricic's class, one of our lead questioners so far in the festival today. What chance does the Hawaiian monk seal have genetics? Like, do we know? I mean, its first battle is against the Iberian ribbed newt, which I've got to say, of all the options, seems like one of our lowest bar easy victories. What do you think?
Yeah, you know, I don't know in terms of that. You know, from a perspective of conservation, we're always concerned about diversity levels. So that'll be something that we'll research to learn more about. You know, when we work on our genetics fun facts for the battles. So yeah. I know seals have a lot of cool adaptations, related to blubber and fat, right? So keeping warm in cold climates, but the monk seals are in a slightly warmer place, even though the waters can be cold.
I think, though, on the blubber note, we've been talking about the walrus lichen battle a few times today. The walrus's blubber is going to just, no matter how much lichen attacks it, it's just going to be able to fend that right off. Now, speaking of a sort of lack of diversity, Eduardo, as a human specialist, I'm really curious. To my knowledge, we went through a big genetic bottleneck at some point in our history, right? So, what does that mean for people who have no idea what a genetic bottleneck is, and what did that do to us? I'm curious.
So when populations leave a continent and go to another continent, you have a reduction in the population. That it's not all the population that was in the previous continent that goes to the next, right? So you have some reduction of diversity there, not only because of the numbers, but there is some processes that we call genetic drift that happens more in small populations. So it tends to make the genetic diversity disappears. But the other thing is that once the population goes through this bottleneck and it populates the other continent, it also develops new genetic diversity there because of adaptation, for example, or new mutations that happen there. But there is always this process of losing diversity and gaining diversity again. But populations in general, each population will be different from each other in the levels of diversity and so on.
Yeah. So one of the ways that I've heard about it that I really love is the idea that a regular band of chimpanzees, 40, 50 chimps, has more diversity than all of humanity, or huge swaths of humanity, because they didn't go through this bottleneck. They're a smaller population now, but that's wild to think about.
Yeah, I'm not sure. I study a few dogs and wolves, dog breeds and wolf populations as well, and they're pretty low diversity because of interaction with humans.
Yeah.
So, the Royale wolves, for example, they are even the Ethiopian wolves. They are a tiny population. They are really low diversity. We studied them, and so there are many wild species that have really low diversity, and because of the interaction with us, and dogs also sometimes you see that.
Yeah, I'm so glad you mentioned Isle Royal wolves. I've almost never heard of that in any broadcast ever. They're so cool. Everyone should finish this broadcast. I've already told people what to do when they finish the broadcast 7 times in 3 programs, which is way too many. I've told people to get outdoors and go exploring. I've told people to fill out their bracket, but also go look up Isle Royal. This is one of the coolest ecological places on this planet, because you have one predator, you have one prey thing, it's very cool. Check it out. Elinor, okay, I've got a question for you from YouTube. Okay, so we were talking about genetics, we're talking about all this cool stuff, and Caden wants to know what happens if DNA breaks during a study. You've got some DNA, it just rips in half. Do you just cry, or do you try again, or do you have a lot of DNA, or what's going on?
Well, let's see. So if DNA broke in a tube, that usually isn't going to matter. So when we sequence DNA, usually we think about how DNA is functioning inside of cells. When you take the DNA out of a cell, it breaks into pieces really fast, and mostly we just go ahead and sequence it anyway, and then try to put the pieces back together again after we know what the sequence of the DNA is. But the funny thing about DNA or genomes, so the DNA that makes up the human genome, there's about 3.1 billion letters of it. And as far as we can tell, 85% of it, it doesn't really matter that much what it looks like. It could change and mutate, and it doesn't seem to have any effect on anything. And so whether the DNA keeps working or not after you break it depends an awful lot on where the changes happen and whether they're changing in a place that's important. And a lot of what we spend a lot of time doing in research sounds really tedious, because all we're trying to do is figure out which positions in the DNA are actually doing something, not even what they're doing, but just starting the search. Out of these 3 billion bases, where should we even look? And we don't actually know that yet.
It's crazy. It's crazy that there's so much exciting stuff going on in the field, and yet that we still have so much ignorance in the most literal sense about this field. It's a very, again, I've been saying this all day long. It's the most exciting time to be curious in science ever. Never mind any other stuff going on in the world. It's just so, if you're fascinated with the world, with the cosmos, with nature, It's the best. And there's programs like this that get people so inspired and excited. And I think it's such a beautiful thing. So Elinor, thank you for that. By the way, I must say, the geneticists, you guys are the most chatty bunch in the private chat of all our scientists so far by like a huge margin. I don't know what's going on. You're just like really excited sending all the stuff, but it's been great. We've been sharing some of the links to some of those great resources in the chat. And Eduardo mentioned that Anne might have an extra thing to add about our ancient DNA, broken DNA. So, pressure's on you.
Right. So you know, as Elinor said, we have 3.1 billion bases of DNA, and it's arranged in our chromosomes, right? And so if you had a really good sequencer that could do long reads, you could actually potentially get a whole chromosome, right? But with ancient DNA, the DNA is shredded, it's got damage, so it's like the most grody DNA possible. It's just, and so it makes it really difficult to put back together. So we often are dealing with really small fragments that we're having to try to piece together and make sure that they're not damaged. So Jurassic Park, it's like probably not going to happen, sadly, because once you get really far back in time, dinosaur far back in time, it's just tiny bits that have chemically degraded.
But it's like scientific detective work with this broken DNA, which is so cool. Like you're piecing together a puzzle that is like thousands, if not a million years in the making. By the way, so I'm trying to come up with like a slogan for each session. Like we're March Mammal Madness 10 a.m. right now, but most grody DNA imaginable is our new title that we're going to do at the end of this broadcast. Our one in the behavior one was the graphs with the snack bars of the desert, because everything eats them. But we're doing pretty well so far. I'm going to take this question to you, and then time flies when you're having fun. We're going to wrap up not long after that. So, Eduardo threw in this phrase so casually, genetic drift. And they want to know, what is genetic drift? What's going on?
Oh, boy. Oh, gosh. You're asking me a question that I should know, like, spot on, just like that. This is an excellent question. And effectively, I view genetic drift as just random processes of change over time. And sometimes those random changes could be really, really advantageous for a population, and they take off and they become highly, highly common within that population. Sometimes they do nothing and they're just there and they add variation to your population. And sometimes they're bad and it could result in the death of an organism, just depending on what those changes are and where they occur in the organism's genome.
So it's like the genetic equivalent of luck. Like there's things that change in the DNA that are adaptive, or everyone's nodding. That's a good sign. Thank you, other geneticists. So like it's adaptive. It's a positive thing or it's a negative thing. With genetic drift, it's a fluke. And who knows where it leads? And it could lead to absolutely nothing. So like, is there an example of this actually for our speakers? I'll bring everybody back in. Offhand, is there an example of genetic drift in any of the species you work on?
So we use genetic drift in my research, for example, to understand how different two populations are. So as time goes, the different populations that are isolated, they start to drift to different directions. So they start to accumulate, or let's say the mutations start to change frequencies, become more or less common in the population, but in different directions. So we use genetic drift to see how divergent are two populations, for example.
Yeah. So Elinor, you put this beautiful thing in the chat about genetic drift, and I love it. So it's our thing that most scientists can't get their heads around. This is what happens when you put things in the chat. I have the power to bring them up as banner, so that's on you. This is the essence of science, though, and I love this. I love when we've talked about this in all our broadcasts today. The fact that students consistently stump scientists with their questions. The fact that there's so much still to learn. The fact that concepts that not all this is easy. Genetics is probably the most difficult topic we're going to be doing today, which is very exciting. We've had the chance to dive into some of the amazing species you guys have featured. We've dove in with some of the essence of this as a field, which is really exciting. So for our audience, thank you guys for such great questions. And everybody, time flies when you're having fun. We are like at the end of our broadcast. And in fact, we might finish up slightly faster than the first two, which means that we all get a gold star at the end of this and win our own personal festival brackets. What I want to stress to everyone today is we've covered questions about how you can learn more. We've covered questions about all the exciting things going on in the field. For the March Mammal Madness tournament, I encourage everybody, head to social media, share your pics, share your excitement, get thrilled, talk about the festival today, share pictures of all of us amazing scientists and then me, the Batman, on camera. Go to the LibGuide. There's resources there for educators, kids of all ages. Check those out. There's so much to learn, so much to discover. Print off your bracket and maybe be like our geneticists and pick bats and things that are clearly never going to win. But because they, sorry. I'm in trouble. No, I mean, we'll see. We don't know.
The thing with March Mammal Madness is that the battles are happening in March. So sometimes some animals will have a disadvantage in March. We don't know, maybe a behavior or maybe an environmental disadvantage. So if an orca and a lion are fighting, but it's in the water, we know maybe he's gonna win, right? So we never know what's gonna happen.
Thank you for throwing in maybe into the orca lion in the water fight. That's very equivocating of you. I appreciate that. I didn't know they were happening in March. That has not been mentioned yet. That's very cool. Well, for what it's worth, amidst all your picks, I clearly have picked the winner, and I'm going to harp on this all day long. Team Sentient Pinecone, the pangolin. It beats anything we talked about. Bats can come at it. Seals. I don't care how much tuberculosis they have. The pangolins are going to win. They got the scales. They're immune. They're the cutest. So lion's going to come running at a pangolin. It's going to see it. It's going to be like, oh, I can't eat that. It's too delightful. And it's going to walk away, which means the pangolin wins. That's my pick. You guys on YouTube, feel free to share in the chat. But Eduardo, Jessica, Elinor, Anne, thank you so much for joining us today. I really appreciate all your insight. And we'll end the broadcast there. Have a wonderful day. Keep up the amazing work. And good luck with your brackets, everybody.